and we'd like to welcome Dr. Marla Spivak. Marla, thank you for joining us today.
Hi, how are you doing?
Really good. How are you doing?
I'm good. Jonathan gave me an excuse to put up the background.
That's beautiful. I just love the fact, I love the group of speakers we have at this year's conference. I mean, you guys all know each other. And, you know, Zoom has this thing where we can put you all in the same virtual room together. Maybe we'll try that in the next conference. Actually, we've been trying to get you to come back to New Mexico for the longest time, and Zoom has kept us from doing that, but we certainly hope to have you in the future. And so if you don't mind, I'd like to start and share with folks that may not know you as well as we do a little bit about your background. And so I have a little story here, and you're going to have to tell me if it's true or not, because someone here in New Mexico says that when you were a bored teenager, you picked up a book about bees in the library and became instantly fascinated by bees and the people who loved and cared for them. I guess it's probably a good thing that you were born when you were born, because you went to the library and read a book. You weren't watching TikTok videos. You know, you actually opened a book. So that's amazing. So that the story goes that you went to work for a commercial beekeeper here in New Mexico, of all places, and you've been known to have said you never looked back. So Marla is a MacArthur Fellow and McKnight Distinguished Professor in Entomology at the University of Minnesota. She's particularly well known for her breeding lines of honeybees that detect and remove disease larvae and pupa, which is called hygienic behavior. I know you were on earlier. Weren't those electron microscope pictures of Varroa just unbelievable? Yes. Anyway, yes. So you've been intimately involved with helping us develop lines of bees that can actually bite the legs off those mites. Marla was instrumental in setting up the first bee tech transfer team in the US, which continues to help honeybee queen breeders select for disease-resistant traits. More recently, she's begun studying the role of resins, and that's why we're so excited to have you here today. Resins which bees collect and mix with wax to make propolis. So we all know something about propolis, and you're going to teach us some more things about propolis today. The bees use them as coatings on the inside of their hives as an example of honeybee social immunity. Marla's lab also studies the effect of the surrounding landscape on the health and nutrition of both honeybees and native bees. So Marla, I can't tell you how excited we are to have you here. I hope everything goes smoothly as far as your slides, but we're eager to hear from you. And just we want to remind folks, if you have questions and you want to ask Marla questions through us, put your questions into the Q&A, not into the chat, and we'll see if we have enough time at the end to go through some questions. So Marla, thank you again for joining us.
All right, thank you. Thank you very much. Hello, everybody, and it's a little humbling, but honored. I'm honored to follow Sammy and then Jonathan Lundgren. And then I really look forward to listening to Megan and Julia and Quentin coming up. So I may have to switch to my phone because of another commitment, but I will be listening in and happy to hear everybody. So I'm going to go ahead and share my screen here. And hopefully everybody can see it now.
Looks good.
Okay, great. So I titled this talk, Why Care About Propolis? But really my whole goal in giving this talk is to convince everybody to love this stuff. I know it's sticky. I know it's on your clothes. It's all over everything I own. It'll never come out. But I'm hoping by the end of this talk that you all have decided that this is such good stuff for bees that you'll tolerate it. So I'll get going here. Honeybees and other social insects like ants and termites and wasps, these are all my favorites, those insects that live in really densely populated societies, have to keep their colonies healthy. And now through COVID, we're all learning what it means to live in really densely populated societies when a disease, or in this case, our case, a virus moves through. How do we keep ourselves healthy? And I won't go into any politics of this. I promise to stay away from it. But the way we do that is the same way that social insects do. And so social immunity is a way that they keep themselves healthy and prevent disease transmission. And this term was coined by Sylvia Kremer from Austria in 2007. And this is how she defined it: the behaviors and organizational strategies and physiologies that social insects use to defend themselves against parasites and diseases so that they can stay healthy. So bees have individual immune systems. If you go inside of a bee, they have very sophisticated immune systems. They do not produce antibodies. So their immune system differs from ours in that respect, that they don't have that type of a long-term memory, but they do have cellular responses, and they have what we call humoral responses. In the cellular ones, they have cells that can engulf and encapsulate foreign intruders, microbes. And humoral responses are the production of these proteins that can eat up, recognize, and then eat up microbes, foreign microbes, or they can melanize them and create a wound response around them to wall them off. But what I like to study is what happens when all the bees come together. All these little individual bees with their individual immune systems, they start collaborating as a colony, and they have behaviors that they use to prevent this disease transmission that support their individual immunity. So simply the division of labor, so segregating by age somewhat, helps prevent some disease transmission. But the two behaviors that I've been studying for quite a while now are hygienic behavior, which I really won't talk about today, but I will talk about resin collection, or propolis, and how and why that's a form of social immunity. So there it is. There's a bee with resin or propolis on her back legs, and I'm going to define resin and propolis shortly. So bees, it's fascinating that they can recognize plants that produce resins. Resins are highly, highly antimicrobial, and they're produced by only some plants as a plant defense. So some plants secrete these resins as a form of keeping themselves, as a form of keeping the plant itself healthy. Because they're antimicrobial, they're a defense for the plant, and they also prevent herbivory. So herbivores don't like to eat these resins, and they can protect against UV rays for the new leaf buds on some plants. And honeybees are going to these plants and they're scraping the resin off of, usually it's on the leaf, a growing leaf. They scrape the resins off and then they pack them on their hind legs. Like you can see in this second photo, this bee has a bright yellow load of resin on each of her hind legs. They carry this resin back to the nest. It's so sticky that the foraging bee can't remove the resin herself. So her nestmates come to her aid and help with their mouth parts. They pull the resin load off of the bee, and then together they cement it, the resin, into the nest cavity. The bees are not changing the chemical composition of the plant resin. Sometimes they add beeswax to it in more or less quantities, depending, but pretty much the resin chemically is exactly the same as it is from the plant. And so you can call it resin or you can call it propolis. What happened was that people discovered this sticky stuff inside the colony and its antimicrobial values, its value for human health, and they called it propolis before they understood that it was derived from plant resins. So resin actually equals propolis. Resin is on the plant, propolis is the resin once it's inside of a colony. And if bees nest in a tree cavity in their natural nesting site, and this is a tree that was brought into the lab here many, many years ago, the colony was dead and we opened up the tree and removed the combs, as you can see on the right. We wanted to know where bees put the propolis naturally because we had read from Tom Seeley's book in 1985 that within this tree cavity, they completely line the interior of the nest cavity all around the honey and the brood with this layer of propolis that he called a propolis envelope. And when we opened up the tree and you look inside, sure enough, here's where the combs were. You can see remnants. But this kind of brown shellacked area is where they deposited a fairly thick layer of propolis. And this red dotted line is where the combs ended, and you can see some mold and other things growing in the tree cavity and not below it. And right there pretty much tells you the whole story. The propolis is killing off these fungi and bacteria and other microbes that are within the nest cavity itself. So on the left is another picture of that same tree, and it's looking out the front door as if you were a bee looking out your front door, and all the thick layer of propolis that they put particularly around the entrance. These were the combs here, but this can be up to a quarter, sometimes ½ inch thick, very thick. In contrast, here's a picture of the inside of our beekeeping equipment. And because the wood is plain smooth, bees are not forming a propolis envelope within our beekeeping equipment. Instead, they do continue to bring in propolis, but they're using it, they put it everywhere where it gets in our way as beekeepers. So they put it in cracks and crevices, the corners of the box, or any cracks in the equipment, under the frame rests, making it very difficult to get frames up sometimes, between the boxes, if there was a box on top of this. That's why we need a hive tool often to get the boxes apart. So I hope to show you that we have made a mistake in using smooth planed beekeeping equipment. It can be planed outside and painted, but on the inside, it really needs to be very rough. And I'll come back to this, how to do that, at the end of the talk. So stay tuned on that. I became really interested in propolis collection for a number of reasons, but what was really interesting to me is that bees are collecting this stuff. They fly out and they find it. They're not eating it. So it's not giving them a nutritional reward at all. And it's energetically demanding because they have to go find it and then they can't get it off their legs themselves. So we wondered, even though we've had in the United States over 100 years of kind of selection against colonies that produce propolis, they're still collecting it. So it must be good for something. And I was really curious why. And this photo here shows you a difference between a resin load and a pollen load of about the same color. So pollen is kind of fuzzy, and resin loads are very shiny, and they're irregular shaped, can be kind of sharp looking. So over the years now, we've been addressing these questions, and at the bottom are pictures of students that have gotten masters or PhDs from answering these questions, and I'll talk about them more as we walk through it. Where are the resins? Where are the bees collecting resins in our area in Minnesota? And what's the benefit of having a propolis envelope? Does it reduce disease load for the bees? Because it is so antimicrobial, does it help them fight off disease? And because it's antimicrobial, how might it affect their beneficial microbes, their microbiome? And then do colonies social medicate? In other words, if they're sick, do they go out and forage for more resin, for more medicine? So to answer the first question in one slide, this is the work of Mike Wilson, who got his PhD working with plant chemists. And hate to reduce all his work down to one slide, but using LCMS, which is liquid chromatography followed by mass spectrometry, he would compare the chemical profiles of resins that he collected from leaves, and then with the chemical profile of resin from the back leg of a bee. So he climbed many, many trees, many species of trees, and got leaves or bark from plants that produced resin. And then we collected bees at the entrance of colonies returning with a resin load. And he would compare these metabolic profiles and found that in our area, bees are collecting almost all of their propolis from plants in the genus Populus, which is mostly eastern cottonwood in our area, balsam poplar, hybrid poplar. Aspen, which we don't have too much in our area, is also from that genus. So I don't know what they're collecting in your area, maybe from cottonwood also, but in our area and it matches the reports from Northern Europe, where the bees in Northern Europe are also collecting mostly from Propolis. So here's Mike Simon Finstrom, who did the initial work, and this was back in 2008 and 2009, so it's been a while now. We needed to create colonies with a propolis envelope, but in beekeeping equipment. And so our first method, which I'm going to say I don't recommend to you as beekeepers, this is an experimental design thing. We collected propolis from colonies, we dissolved it in 70% lab grade ethanol and made a tincture, an extract. And we made a 15% extract and we painted that in the inside of 5-frame nuke boxes. So the picture here on the left is just the ethanol, the alcohol, as the control we painted on. It evaporates right off, but we needed to know if the ethanol had any effect on the bees. The one in the middle was some propolis we got from Brazil. It's a green propolis from the genus Baccharis, which includes rabbit brush, which might be in your area. And the one on the right is propolis from Minnesota, which comes in a little bit red from the populace. So we made these, we painted on a propolis envelope, and then we put bees and frames and everything in them for our experiments, which I'll talk about shortly. But that's just the first way that we made a propolis envelope. Then later, oh, I didn't want to put that there. Maybe I do. Another way was to use a propolis trap. So maybe you've seen these, they sell them at beekeeping supply places. It's a grid that you place over the colony and it has these 7mm gaps. So that's how wide they are. And a whole grid is about 10mm deep. So it's a little thicker than the gaps itself. You put it on top of a colony, and the way we do it is to take a wooden rim with holes drilled into it. And then we put the lid on over the rim, and air and sunlight comes in through these holes, which bees do not like. And many colonies quickly fill in the gaps with propolis, which you can see here. So we knew that these propolis traps are a great way for bees to collect propolis and for us to trap it. So what we did, what Renata Borba did, a different student, was to take those propolis traps, which are a little bit expensive, but it's research, and she cut them up and stapled them within bee boxes. Okay, so you can see on the left, a propolis trap stapled in, and on the right, if we remove the trap, what that quote propolis envelope looked like. But for our experiments, we kept the trap in. I also don't suggest you do this because it really interferes with the bee space and it makes beekeeping very difficult. But through these experiments I'm going to be talking about, these are the two ways that we made a propolis envelope. So in one experiment, Mike Simon Finstrom decided to make the bees sick with chalk brood, which is fairly easy to do. You collect some chalk brood mummies, fungal mummies. The bees kick them out of the front of the colony, and you can collect them, homogenize them, and mix them into pollen patties, pollen supplement, and then feed it to the bees. And colonies within 2 weeks will develop symptoms or signs of chalk brood within the combs. So then we had colonies in these nucleus boxes where we painted the propolis envelope on there, or like varnish. And then we made them sick with chalk brood, and then we went in and counted the number of chalk brood mummies in the combs. And if they had a propolis envelope, the number of mummies they had was on average about 15 in 6 colonies. And the other 6 colonies that did not have a propolis envelope on average had 108 mummies. Okay, wide variation, but highly different, highly significantly different. And this tells me that, okay, the propolis did not cure the disease, but it really helped them reduce the disease load. And it's important for you to understand, this is not a treatment. This is something that really, really helps the bees maintain their own health. In another experiment that Renata did, and this time her setup was with those propolis traps stapled inside, she made colonies sick with American foul brood, which I also don't recommend you do, because at the end of this experiment, we had to burn all the colonies. She'd made a sugar solution, And she mixed in spores, bacterial spores from Pennybacillus larvae, which is the bacteria that causes American foul brood disease. And she sprayed this sugar bacterial spore solution on every comb and gave them to bees that had a propolis envelope, in that case in this kind of maroon colored bars, or that did not have propolis in the gray bars. And in this case, instead of counting, we gave it a score of 1, 2, or 3. 3 is really bad. 2 was very bad. You know, if you look at one comb, there were over 20 cells filled with of brood cells with American foul brood, and 1 was 5 cells or less per frame. And you can see by the end of the season, again, it did not cure the disease, but colonies with a propolis envelope, the severity of their American foul brood was much less. It was 1 versus over 2 on average. So this is saying, okay, this stuff is helping them fight off diseases. That's good. We also, over and over, looked at the immune response of bees. And I'll explain our results here. I'm showing you Renata's results where she used those propolis traps. And we marked bees when they were just born and then took them out of the colonies when they were 7 days old, and then measured their immune response by looking at using quantitative PCR. You can ask me later how that works, but you can measure the amount of proteins that the bees are producing as part of their immune system. And so these names across here, hymenoptacin, abacin, defensin, et cetera, are some of those proteins that bees produce as part of their immune system, and relish and phenol oxidase are other immune-related responses that we can measure. And every time that we measured bees that were in colonies with a propolis envelope, their immune response was lower compared to bees from colonies without a propolis envelope. So if you look at the maroon bar, it's lower than the gray one, lower than gray. Even whether they're pointing up or down, it doesn't matter. I know it gets confusing, but lower. At first we thought, whoa, that's weird. We also found, but I'll explain it, but we also found that by measuring their immune systems over the season, bees in colonies with a propolis envelope, their immune response was less variable. It didn't go up and down and up and down. They weren't activating and then reducing their immune systems. It was pretty stable and consistent over the season. And so we were thinking, wow, we think this is a good thing, because when bees have to activate their immune systems, it's at a cost to them. It's at a cost to you to activate your immune system. So having a propolis envelope reduce the investment bees invest in their immune response, because the antimicrobial properties of the propolis reduce the opportunistic and pathogenic microbes. So it was getting rid of all of those other microbes in the nest cavity, so the bees did not have to activate their own immune systems. And here I always give a human analogy to help you understand how this works. So I have an allergy to mold. And if I'm sitting in a room full of mold, I'll start sneezing. And if you measured my immune system at that point, it would be highly activated. But if I would take a propolis tincture and paint it, or if I would collect propolis and cement it all, make a propolis envelope in the room that I was in, it would kill off the mold. And as a result, my immune system would be quieter. So that's exactly what's going on for the bees. The propolis envelope is killing off all these other microbes in the nest cavity, so that the bees are not having to activate their own immune response, which is a really, really good thing for bees. Okay, so we know it's antimicrobial, but like I said, there are bacteria in and on the bees that are beneficial to them. And so if propolis is really antimicrobial, what happens to the microbiome? And we did 2 studies on this. One of them with Renata, who sent bees to the USDA Bee Research Lab in Baton Rouge where they dissected these bees and sequenced all the bacteria they found in the abdomens, which include the gut. So here is a diagram of the bee's guts, you know, the crop, the ventriculus, the intestines, the rectum, and each area of the gut hosts its own community of beneficial bacteria and fungi, although we were only measuring the bacteria in this case. So each of these areas has a known assortment or community of microbes that it needs for survival. And we know that bees are not eating propolis directly, but I'll come back to that. So they're not eating the resins. Just the exposure to the propolis, we found that the core gut microbes were more abundant, and the other opportunistic and pathogenic microbes were less abundant when bees had this propolis envelope. We repeated this in another way. And in this case, we only looked at the mouth parts. And it may sound kind of funny, but bees and all organisms have microbiome in their guts, but we also have microbiomes outside of our bodies and everywhere in our mouths. And if you think about it, bees get their mouths into everything, right? So they feed larvae with their mouth parts, they collect nectar, and you know, they're visiting flowers with their mouth parts, they're sharing food inside the nest through trophallaxis, they're feeding the queen, they're doing everything with their mouths. So we thought, let's look at the proboscis and the mandibles, the mouth parts of the bees, and what happens to the microbiome that's on these locations. So this was work by Holly Waldallenberg down here in collaboration with Kirk Anderson, who's at a different USDA facility, Bee Research Lab. This one's in Tucson. So Holly collected these bees and froze them and then snipped off, I know it sounds horrible, but they were dead. She sniffed off just the mandibles here and the mouth parts and froze them. And we extracted the DNA, and then Kirk Anderson sequenced it. So here's a graph. Sometimes they call them seizure graphs, because if you, I don't want you to have an epileptic seizure, but anyway, you can just, each of these rows going across is an individual bee. And the ones on top are bees from colonies with a propolis envelope. And the ones on the bottom are from colonies, bees from colonies with no propolis envelope. And the different colors are different bacteria that we identified just on the bees' mouth parts. So the first thing, you can just look at that and say, okay, the mouth, there's a lot more bacteria on the bees without a propolis envelope compared to bees with a propolis envelope. And these three core species, shown in blue, orange, and dark blue, Bombella, Lactobacillus, and Fructobacillus, misspelled, needs a C right there. You can see they increased in abundance. There were more of them if there was a propolis envelope. And when we identified the other bacteria, which are shown here, now in blue here, Pseudomonas, Pseudoaltomonas, Streptococcus, these are all bacteria, these are all pathogenic or other bacteria that can cause disease, but not necessarily. And you can see in bees with no propolis envelope, there were a lot more of these other bacteria compared to bees with a propolis envelope. And here are the three core mouthpart microbiome species, Bombellus, Lactobacillus, Fructobacillus. Those are the three you can see in higher abundance. So the propolis is actually supporting the growth of the beneficial bacteria and killing off or reducing the number of those other disease-causing bacteria. In all of our experiments, we did not find that the propolis envelope lowered varroa levels, too bad, nosema or viruses. But other researchers have seen when they fed nosema in solution that it reduced nosema. I don't really recommend feeding propolis to bees in solution as a practice because it is such a strong antibiotic. And more recently, um, this is funny. Here we go. My slides are skipping ahead a little bit, but there we go. More recently, or just recently, this article was published out of Italy from Francisco Nazzi's lab, which is really, he's a great chemist and they do really great work in this laboratory. They found that the bees were applying a small amount of propolis on the cells in brood comb. This is a picture from that wild bee nest that we had, and toward where the combs touched the wall of the tree, we could see this very, very thin layer of propolis on the rims of the cells. But these authors found that there is some propolis, not very much, very, very small amount of resin within the wax cells in the brood nest. And they could identify the compounds in them, and they were from propolis. They could see some of the propolis compounds in the wax. Some of them were not present, others were really, really concentrated. And they did this really cool thing. They took these gelatin capsules here and they applied very, very small amounts of resin, the same amount that they would see in a wax cell, into the gelatin capsule. And they transferred 5th instar larvae into the capsules. And then, whew, wow. Okay, they put these larvae in there and then raised them inside the laboratory in an incubator. And they put varroa mites in some. So they had, they were rearing these pupae in gelatin capsules with and without propolis compounds and with and without varroa. And what they found is that when the pupae were reared in brood cells with a little bit of propolis, it killed it, the mortality of the mites increased by about 20%. So small effect on varroa, killing off varroa, and it reduced the fertility of varroa, how many offspring they were produced, by about 50%. So this was super exciting. It just came out. Now let's see if I can go forward one slide at a time. Okay, good. From a different lab, May Birnbaum's lab in Illinois, she also found propolis compounds in honey. So now we're starting to find them in wax and also in honey. And this is a quote from her paper. One of the main phytochemicals, which are plant chemistry, plant compounds, reported in honey, of the main compounds, pinobanxin, pinosembrin, quercetin, chrysin, and galligan, few of these compounds are actually found in nectar. You see them in honey, but not in nectar. And all of these compounds are coming from propolis. See, it did it again. There we go. Although propolis is not known to be ingested per se by the bees, the fact that common constituents of propolis are shared among a wide variety of honeys in many, many geographic regions suggests that ingesting honey may be one way bees are getting the benefit from the propolis-derived compounds. So they're not eating the resin. The honey gets infused with propolis compounds. The wax may be infused, or the bees are directly applying a little bit of propolis on it. And together, it's helping their microbiome, and it's helping them ward off varroa by a little bit, not by much, but a little bit. Okay, the last question. Do bees increase their resin foraging after they're challenged with a fungal or bacterial pathogen? Are they self-medicating, or more correctly, are they social medicating? In other words, are they going out to collect more, quote, medicine, resin from trees if they're sick? And that would help the whole colony medicate. So in order to do this, these were kind of laborious experiments. We would set up boxes, small nucleus boxes. In this time, no propolis envelopes and healthy colonies, no diseases. And the first thing we did was take a small army of us and close up the colonies twice a day for 2 weeks. and sit in front of them and count the number of bees returning with resin on their back legs. We also counted pollen foragers for reference. So if you do this yourself, for every 100 or so pollen foragers, you're going to see about 2 or 4 resin foragers. It's fairly rare. So after we got an average amount of pollen or resin collection for each of these colonies, we challenged them. In other words, we gave them either chalk brood or American foul brood like we did before. And when they were sick, we repeated these resin counts. We closed the colonies twice a day for 2 weeks and counted the number of bees returning with resin. And what we found, I'm just showing you 1 year of data, but we repeated it over 3 years and kept finding the same result, which is just amazing, that if we challenge them with chalk brood, which is Ascosphaera apis, fungal pathogen, this one, this gray bar, they increased the number of resin foragers significantly over the controls, which were not challenged at all, no disease. But if we gave them, made them sick with American foul brood, which is caused by Penibacillus larva, this one, they increased resin foraging a little bit, but not significantly different than the controls here. A, B, and A letters in common mean they're not different, but A and B are really different. So why it is that bees would collect more resin after they're sick with a fungal pathogen, chalk brood, and not with a bacterial pathogen, American foul brood, I have no idea. Maybe you have some ideas, but we have no idea. It's one of the bees' mysteries, which is kind of cool. I like it that bees still have mysteries, but very, very interesting. So in summary, propolis is really good for bees. It provides everyday benefits to their immune systems. It helps promote their core microbiome species. It helps them reduce disease load. It helps them reduce varroa loads a smidgen, a little bit, and reduce chalk brood and foul brood also to some extent. So it's one step toward really good healthcare. If you have a healthcare system, you need multiple approaches, and allowing bees to have a propolis envelope in the colony is one good step forward. So how do we do that? How do you do that as beekeepers? Because like I said, I don't recommend that you collect propolis, extract it, and paint it back on the colonies, because you're going to lose some of the antimicrobial effects from the abstract. Using the propolis traps inside the colony interferes with bee space, and it's a big pain in the neck. So here's Maggie Shanahan, another student, and she, as one of the projects for her PhD, what she's been doing is testing these bee boxes in commercial operations. So these boxes were built by a friend of ours here in Minnesota who mills his own lumber and builds his own bee boxes. And he has made very deep, jagged, rough grooves inside the boxes. So these are about 7 millimeter grooves, which is exactly the same as the propolis traps. And they're 7 to 9 millimeters deep. And they're jagged, as you can see here. It's not a smooth groove here. And the bees very rapidly, most colonies, fill in these grooves with propolis, and it doesn't interfere with the bee space. So here is another shot of those boxes. We had him make a bunch of them. We gave them to a commercial beekeeper where we had 60 colonies in the roughed groove boxes and 60 in just standard commercial smooth boxes. These were in a commercial migratory operation, and we sampled the health of the bees and everything over a couple of years and found that this was great news to us. The propolis increased the population of the bees slightly, instead of before they were going into almonds, instead of 7, they had a little over, maybe up to 8 frames of bees on average. Fortunately, we found that it did not interfere with honey production, because a lot of beekeepers had been asking me, wow, if I make my bees collect a lot of propolis, will it reduce honey production? And apparently not. And again, the more propolis the bees collected, the lower their immune response was. So we were seeing the exact same thing in a commercial operation that we were seeing in our experiments at the university, which was great to see. And we'll be, Maggie's summarizing all this data and starting to write it up for publication now. This is the only data that's not published so far. Okay, I'll come back and thank plants that produce resin. These are cottonwood leaf buds. If you go to a cottonwood tree while it's growing, anytime over the growing season, from very early spring to fall, you break off a branch and you smell it, it will smell like beehives. And thanks to the bees that collect the propolis, to the funding sources, and to my many great students that make me look great, but this is all of their work. And now I'll stop sharing my screen. I've been a crazy driver of PowerPoint, but hopefully you've followed, and I'll take some questions. Thank you.
Marla, thank you so much. You know, we had heard last year about some of this work, and I knew you were, playing with propolis and treating some boxes differently, but the science that you've applied to it to actually get quantitative results is really just quite remarkable. And I'll just say for myself, when I first started beekeeping a few years ago, I knew about propolis, but I thought they were really just using it as a spackle, to seal up cracks and keep them warmer in the winter. But now we all know much more about that. So we do have some questions, but because I'm the host, I get to ask as many questions as I want.
Okay.
So I'm curious, when we do have to scrape some propolis in the process of just moving frames, and I do sort of collect it. I haven't really made a tincture for myself yet or my friends, but is there any benefit for sort of redistributing propolis that you collect with your hive tool into a certain location in the hive, or is it?
I don't think it, well, you can. That sounds like work to me. I will say that propolis over time loses some of its antimicrobial properties. So if it's in your way, you need to scrape some out.
Yeah.
But if you have boxes with rough interior, and the rougher that you can do, the better. If you have these boxes, then the bees will just continue to bring in more the next growing season. So I'm a big fan of letting the bees do the work that they do, because every time I try to do it for them, I seem to mess it up.
Well, I for one can definitely relate to that. Okay, great. So I have this question in addition to Krista Coggins is asking, I wonder if just using rough sawn wood would induce propolis application inside the hive.
It does, but they don't bring, it's not as, the gaps, the crevices are not as deep as within a tree cavity, so they won't bring in as much propolis with just rough wood. We've tried it, as they do when you put actual grooves that are very rough. So think of the inside of a tree cavity, and that's what you're going for. The more, the better. However, if all you can do is rough wood, go for it. It's better than nothing.
So do I think when I've heard this, of some version of this in the past from you, the gentleman that does has created the boxes with the grooves, was there some move afoot for him to actually patent that or commercialize it?
He is selling them, and of course, I can't remember the name of his company right now, and I'm sure it's just a hit on his own, and I don't know if he could get overwhelmed, but maybe while Megan is talking, the next person, I can look on my computer and get his company name.
Great. So, you are somewhat familiar with New Mexico, obviously, but Jim is asking, he says, so native cottonwood, mesquite, juniper, pinon, pine, and elm are all high in resin for New Mexico, which is a blessing, correct?
Yeah, so I don't know anything. Have you ever seen bees foraging resin on mesquite or pinon or the other plants that you were talking about? So I don't know if they're collecting resins from those. You'd actually have to see them on those plants to know if those are resin producing plants.
But definitely are cottonwoods then.
I'm just going to throw in here that Mike Wilson, the student that climbed the trees, he did collect resin in our area from pine and spruce and other conifer trees, but none of our bees ever collected propolis from pine or spruce in our area. It was very curious. I don't know why they go for cottonwood, but they do.
Well, we're lucky to have so many here in New Mexico. Okay, Jim is also asking, is there a breed of bees, Italian, Russian, et cetera, that will gather more resin for propolis compared to other breeds?
Right. So Caucasian bees were notorious for propolis collection. And we introduced Caucasian bees early on in the United States, like in the whatever 1800s, 19, whatever. I don't know when we introduced them, and then stopped bringing them in because they produced too much propolis and beekeepers didn't like it. But I am looking at lots of other bee types right now because we do have another bee breeding program going on. And within, I have some Russians and I have some VSH and some and I have, Italians and Carniolans. And within them, you'll always find colonies that are big propolis collectors. And it is heritable, meaning you can select for this trait, it's genetic trait. So when you see any colony, whatever breed that brings in a lot of propolis, raise queens from it.
Great, great. Let's see, Tina Sebastian from Colorado is asking, what would happen if we sprinkled propolis powder over comb or over frame tops? What about spraying comb and or bees with water tincture of propolis?
I knew beekeepers would do this. Propolis can also be toxic. It's a medicine. It's a very powerful antibiotic. And Maggie Shanahan, the last student that I was talking about, next summer we'll be doing some more lab studies to show how much is too much. So please don't spray it directly on your combs. I think you're going to end up killing larvae. So the bees are putting a very, very tiny amount on there. Please let the bees do it themselves. Thank you.
No, that's great. I'm glad that came up because it's sort of an obvious question, but you put it to bed rather succinctly. That's great. So a lot of people are asking about the roughening process. I know you showed the grooving. More roughening is better. And so, a lot of us have existing boxes, and there's many questions on the Q&A about could we use sandpaper, So I'm guessing any roughening is good, but to really get the kinds of propolis you're talking about, you really need these grooves.
Yes, we've done sandpaper. We've done roughening things up. We put window screen because it wasn't as thick as a propolis trap. We've put burlap. Bees tear down burlap. They tear down any kind of light plastic that you put in there. It takes about 2 years for them to finally get around to putting propolis in window screen. So really the thing that worked the best was to mimic what's going on inside of a tree.
Yeah, that's great. We have one person who, Belladonna, says that he or she, I'm not sure, he or she notches deep lines inside my hives with a chainsaw.
Whoa. Well, you be careful that you don't go.
I know.
You must be better with a chainsaw than I am.
Yeah, no kidding. Okay, so we have a fairly long question from someone that Melanie Kirby. I'm going to read it to you. In our queen rearing mating nukes, we use queen castles, which is a standard Lang box with grooves where we slide in dividers.
Mm-hmm.
The bees always fill those with propolis when the dividers are out. Also, placing a screen on top of the mating nukes to keep queens from crossing into other sections, the bees fill that screen with propolis. I've always described it as a propolis patch that helps to keep their nursery sterile. Plus, then we can pull the screens off at the end of the season and freeze them, and then roll the screens and the propolis flakes off so very easily to harvest and use for other value-added products. And then reuse the screens and the bees refill it with propolis during the rearing season. We even leave on throughout the year, not harvesting all of it. So I guess that wasn't really a question, but do you have any thoughts about that?
No, all of that's great. Yeah, it just shows that those grooves are great. Bees just love to fill grooves with propolis, and screens they will too. So those are just ways that, I always say beekeepers are the most inventive and creative people I know. So if I get this information, the science, the data out there about the benefits of propolis, I have a feeling that beekeepers will come up with really good ways to create rough boxes or to stimulate bees to make an envelope. But I do encourage you to have it in an envelope within the box instead of spraying directly on
Yeah,
the bees or in
excellent,
the homes or feeding.
excellent. Okay, Kathy Grassel asked, many people treat their trees for bark beetles with a systemic insecticide. Will resin collected from these trees be contaminated with this insecticide? And if so, would there be sublethal effects on the bees?
Right. So Mike Simon Finstrom right now in Baton Rouge with other colleagues down there is looking at contaminants within propolis. They're not finding any neonics to my knowledge, but they do see some fungicides in the propolis. Not all of the chemicals that are applied can you see in the propolis, but bees, remember, they're not eating the propolis directly. And when he's done trials in the lab of exposing the bees to the amount of fungicide that he sees in the propolis, it had no effect on them. So I'm thinking the propolis is more like a trap. It's holding those compounds in there, but not necessarily affecting the bees because they're not eating it.
Great, great, thank you. So you showed some really cool pictures of bees with propolis on their legs versus pollen. Patricia Donahue is asking, when looking at bees collecting pollen and resin, it looks so similar. Is there a key thing to look for to see the difference? Some of my bees were bringing in what I thought was pollen last week, but could not figure out where it was coming from. Maybe it was resin.
It could be. It comes in many colors. It could be yellow or red or white or green or gray even. So you can't judge it by its color, but it is shiny to your eye and you kind of catch a glimpse of it and it's irregular shape. So pollen loads are more like dough balls or something and fuzzy, fuzzy looking. They're not fuzzy themselves, but propolis is very shiny. So if you stare at a colony for 15 minutes, you'll see one or two.
Okay, excellent. Let's see, we're just about finishing up here. Many of us do collect some propolis and make a tincture, usually with, I guess, with ethanol. One of our attendees says, are there good propolis extracts you recommend for human consumption?
Yeah, I get in trouble if I go into the human medicine world by my dean in my
Sure.
college.
Yeah.
So don't get me in trouble.
No, I don't want to do that.
But what I will do is put in a plug for a conference coming up in May, and it'll be on Zoom. It's the International Propolis Research Group. And they are researchers from all over the world, and the majority of them study the benefits of propolis for human health. And they report on all of their clinical trials and all the ways they make extracts, either water or alcohol extracts, and all the research that's being done for human medicine. So I highly recommend International Propolis Research Group, IPRG, coming up May 4th, I believe, for 3 days.
Okay, excellent. I think we covered most everything. And I know you've created a bunch of new propolis addicts here, and we won't be so annoyed when it gets stuck on our tool and we'll say, wow, this hive's got lots of propolis in it. I especially liked the part about when you challenged the hive with disease, that the bees are so smart to go out and get more propolis. That was remarkable. I really, I've always, I'm learning more and more how much smarter the bees are than we think they are. And I guess we'll just keep continuing to learn that with your help and the help of all your research colleagues. I just know so many people that have worked with you that talk so fondly about the work that you do and how you work with the other researchers. So thank you for that. We're really proud to have you here today. We hope to hear more from you in the future. So keep up that wonderful work. And thank you for joining us today.
All right. Thanks so much. Bye-bye, everybody.